and welcome. My name is Ottmar and thank you for joining me on my YouTube presentation. I was told to pick a biology role model, someone relevant and relatable, someone who I think is a valuable asset to the biological studies community, and I think that person is Miss Carolyn Bertozzi. You might be wondering what that figure is down on the right-hand side. This is a glycosylation landscape that contains an example of some sort of cancer. This is one of the many things Carolyn Bertozzi worked and still works on as a scientist. Anyways, I'll get more into this later, so first let me introduce the wonderful woman that is Dr. Carolyn Bertozzi. So, what makes up Miss Bertozzi? How did she get here, and was this always her goal? Carolyn was born in 1966 in a place called Lexington, Massachusetts. Science has been a subject of interest her whole life due to her father, William, who was a professor at Massachusetts Institute of Technology. Miss Bertozzi adds that by saying science was the focus of the house. She says that they would wear MIT t-shirts and go to MIT day camp. Talk about commitment. However, Caroline decided that Harvard University in Cambridge, Massachusetts was a better fit for her instead. She believed that the school offered strengths outside of science and entered as an undergraduate in 1984 with no selected major. For a brief time, Bertozzi thought that becoming a music major would work due to her talents on the keyboard, which placed her in many college rock bands. However, she debates this by saying, "I've always centered on the sciences." She began majoring in biology and eventually moved to chemistry, stating that after taking organic chemistry, I fell in love with it. Outside of science, Professor Bertozzi has many incredible attributes as well. So let's talk about what makes her so great. She is an advocate for diversity, more specifically as someone who is part of the LGBTQ community. I really admire her because she's been out since the late 1980s, when being a lesbian could have jeopardized her career. As someone who is part of this community as well, I really respect her for that. Moving on, if you look at the Bertozzi Group logo associated with her, it has something really familiar. You may be able to connect this back to slide 1, which I commented on, and is a visual representation of what Dr. Bertozzi does in terms of research. This is inspired by her first project at Stanford, which designed a method for stripping the sialic acids away from the ends of glycans on cancer cells. For 50 years, researchers have noticed changes in glycan patterns on the surfaces of cells, but Bertozzi was the one that made a huge leap forward in the study. Now let's get into one of her bigger studies, titled as From Mechanism to Mouse. This work in her laboratory led to the development of 2 bioorthogonal transformations that exploit a chemical compound called azide. If you don't know, bioorthogonal chemistry is just any chemical reaction that can occur inside a living system without interfering with their processes. Bertozzi used conjugates labeled into the figure, which led to imaging azide-tagged glycans in living systems. This was the first application of bioorthogonal chemistry to use in vivo imaging, which was a landmark in the field. This experiment worked to find the spatiotemporal changes in the cell surface glycosylation in developing zebrafish. They found that glycans expressed during earlier stages of zebrafish embryogenesis could be detected by direct microinjection of the yolks of single-cell embryos, which was helped encourage new studies in the field. Recently, Bertozzi modified a reaction which resulted in copper-free click chemistry, a reaction that introduces copper into a cell which is toxic. This faster version of bioorthogonal chemistry allowed Bertozzi to track the activity of glycans in cells over time. These discoveries have been used to study how cells build proteins and other macromolecules, develop new cancer medicines, and to produce new materials for energy storage. However, Carolyn Bertozzi currently works as the Anti and Robert M. Bass Professor in the School of Humanities and Sciences and as a professor of chemistry in the Stanford University. Thank you so much for watching. I hope you found an interest in this amazing female scientist and you were able to advance your knowledge a little more on her topic of study. Feel free to look into Dr. Bertozzi more because this presentation does little justice for her immense accomplishments. Here are some sources of everything I covered. They go into a lot more detail and are great if you want to learn more about her and her studies. Only 6 were used, but trust me, there is a lot more out there. The following slide also has links to every picture used in the slideshow if anything else piqued your interest. Thank you again for watching and have a nice day.